Toxins and growth regulators. Introduction. Toxins and growth regulators play a very important role in plant pathology. Plant toxins are toxic secondary plant metabolites which occur naturally in food, feed, weeds and ornamental plants with tremendous chemical diversity. Non-host specific toxins. Tab toxin. Tab toxin chemically is a dipeptide composed of the amino acids threonine and tabtoxinine. This toxin plays a role in the wildfire disease of tobacco. In this disease, leaves exhibit necrotic spots surrounded by a yellow halo. Identical symptoms of the disease may be induced by culture filtrates of the organism or purified toxins with symptoms identical to that of wildfire of tobacco. Since similar effects may also be observed on a relatively wide range of hosts, the toxin is non-host specific. The inhibition of the enzyme glutamine synthetase is the primary mode of action of the toxin. Fasciolotoxin. Chemically, the toxin is a tripeptide of ornithine alanine arginine with a phosphosulfonyl group. Within cells, the toxin is enzymatically cleaved, releasing phosphosulfonyl ornithine, which is the toxic moiety. Cellular effects are a result of the inactivation of the enzyme ornithine carbonyl transferase. This is the toxin involved in one of the bacterial bean blights called halo blight. Symptoms of the disease incited by the bacterium can be produced by the toxic alone. 10 toxin. The toxin is a cyclic tripeptide that binds to and inactivates a chloroplast coupling factor protein involved in energy transfer and also the inhibition of light dependent phosphorylation of ADP to form ATP. This is the toxin produced by Alternia alternata. The disease induced by this organism is primarily a seedling disease in a wide range of plant species. Seedling death results when greater than 1/3 of the leaf area becomes chlorotic and reduce vigor with less than the amount of leaf chlorosis. Host specific toxins. When one reduces the toxin theory to its most elementary form, it can be stated that all of the symptoms of a given disease result from the direct action of a toxic product of the pathogen in that disease. Some of the following are criteria which support the theory. Number 1. The toxin, when applied at concentrations which could be reasonably expected in or around a diseased plant, produces in a susceptible host all the symptomatic characteristics of the disease. Number 2. The pathogen and the toxin exhibit similar host specificity. Number 3. The ability of the pathogen to produce the toxin varies directly with its ability to cause disease. Number 4. A single toxin is involved. The first toxin isolated that fulfilled the above criteria was that produced by Coccilobolus helminthosporium victoriae. Helminthosporium leaf blight of oats appeared in 1945 with the introduction and widespread use of the oat variety Victoria and its derivatives which contain the VB gene for resistance to crown the rust disease. In the Victoria lines, the fungus which infects the basal portions of the plants, and as it infects, it releases a toxin that is carried to the leaves, causing a severe leaf blight and death of the plant. All other oat varieties and other plant species are either immune or sensitive to the toxin was proportional to their susceptibility to the fungus. In addition to the toxin producing external symptoms identical to that induced by the pathogen, internal biochemical and histochemical results induced by the toxin were identical to that induced by the pathogen. Chemically, the toxin is a complex chlorinated cyclic pentapeptide. Ultrastructural studies demonstrate that primary target of the toxin is the plasma membrane. It apparently binds to proteins, and in some cases it is yet to be determined if the mechanism alters the metabolism of the susceptible cells. T-toxin. This is another example of a host-specific toxin. The toxin is produced in common corn disease, which is the southern corn leaf blight, by the fungus Coccoliobolus heterostrophus, formerly known as Helminthosporum maedis. T-toxin is produced by race T of the fungus that first appeared in the United States in 1968. The toxin chemically is a mixture of linear, long polyketols with 35 to 45 carbon atoms. The toxin specifically affects mitochondria of susceptible cells where ATP synthesis is inhibited. HC toxin. An example of a host-specific toxin is the HC toxin produced by the fungus Coccoliobolus carbonum, which incites leaf spot disease in corn. The toxin is specific to only certain maize or corn lines. The mechanism of action of this toxin is poorly understood at this present time, although the molecular and biochemical basis for the resistance is well understood. These are some of the toxins which play a role in a plant disease. Let us now look at what is called as growth regulators. Plant growth is controlled by a group of naturally occurring compounds with hormonal action that are referred to as plant growth regulators, plant growth hormones, etc. These regulators can be natural or synthetic. The major group of natural plant growth regulators are the auxins, gibberellins, kinins and the ethylene. Plant growth regulators are just like any other hormone, that is, they act in very small concentrations and with slight changes in the normal level, resulting in profound changes in the growth pattern of the plant. Pathogens often cause an imbalance in the hormonal system by causing the infected plant to produce more or less hormone, or in some cases the pathogen itself alters production of these hormones, thus changing the hormonal levels. Some of the commonly observed symptoms related to effects on plant growth regulation are stunting, overgrowths, scaling, root branching, adventitious root formation, defoliation, rosetting, leaf epinasty, etc. Plant growth regulators, which are also called as plant hormones, are numerous chemical substances that profoundly influence the growth and differentiation of plant cells, tissues and organs. Plant growth regulators function as chemical messengers for intracellular communication. Auxins, gibberellins, cytokinins, abscisic acid and ethylene work together, coordinating the growth and development of cells. In addition to the 5 principal growth regulators, 2 other groups sometimes appear to be active in regulating plant growth, which are the brassinosteroids and polyamides. Let us now look at abscisic acid. Abscisic acid, also called the ABA, is one of the most important plant growth regulators. In plants under water stress, ABA plays a role in closing the stomata. Soon after plants are water stressed and the roots are deficient in water, a signal moves up to the leaves, causing the formation of ABA precursors there, which then move to the roots. The roots then release ABA, which is translocated to the foliage through the vascular system and then modulates the potassium and sodium uptake within the guard cells, which then lose turgidity, closing the stomata. Abscisic acid exists in all parts of the plant, and its concentration within any tissue seems to mediate its effect and function as a hormone. Its degradation or catabolism within the plant affects metabolic reactions and cellular growth and production of other hormones. Plants start life as a seed with high ABA levels. Just before the seed germinates, ABA levels decrease. During germination and early growth of the seedling, ABA levels decrease even more. As plants begin to produce shoots with fully functional leaves, ABA levels begin to increase slowly down cellular growth in more mature areas of the plant. Stress from water or predation affects ABA production and the catabolism rates, mediating another cascade of effects that trigger specific responses from targeted cells. The next hormone is the auxins. Auxins are the major growth regulator in plants and are defined as growth regulators whose major mode of action is cell elongation. They resemble indole 3-acetic acid in their activity. The naturally occurring auxin is indole acetic acid, which is the IAA. Auxins are compounds that positively influence cell enlargement, bud formation and root initiation. They also promote the production of other hormones, and in conjunction with cytokinins, they control the growth of stems, roots and fruits and convert stems into flowers. Auxins were the first class of growth regulators discovered. They also affect cell elongation by altering the wall plasticity. They are responsible in stimulating cambium, which is a subtype of meristem cells, to divide, and in stems cause secondary xylem to differentiate. Another function of auxins is to inhibit the growth of buds lower down the stems, which is the apical dominance, and also to promote lateral and adventitious root development and growth. Leaf abscission is initiated by the growth point of a plant ceasing to produce auxins. Auxins in seeds regulate specific protein synthesis as they develop within the flower after pollination, causing the flower to develop into fruit to contain the developing seeds. Increased levels of auxin are seen in plants infected with fungi, bacteria, viruses, molluscs and nematodes. Following are a few important diseases directly related to the altered auxin levels. They are corn smut, club root of crucifers, southern bacterial wilt, crown gall. These are some of the good examples of diseases where altered auxin levels have been implicated in the disease. The next hormone is the gibberellins. Gibberellins play an important role in seed germination, affecting enzyme production, which is responsible for food production used for growth of new cells. This is done by modulating chromosomal transcription. In grains such as rice, wheat, corn, etc., a layer of cells called the alluron layer wraps around the endosperm tissue. Absorption of water by the seed causes production of gibberellins. The GA is transported to the alluron layer, which responds by producing enzymes that break down stored food reserves within the endosperm, which are utilized by the growing seedling. These hormones promote flowering, cell division, and in seed growth after germination. Gibberellins are also responsible for bolting of rosette forming plants, increasing internodal length. Another function of gibberellin is to reverse the inhibition of shoot growth and dormancy induced by abscisic acid. About a century ago, a rice farmer in Asia noticed some exceptionally tall seedlings growing in their paddies. Before these rice seedlings could mature and flower, they grew so tall and spindly that they toppled over. In Japan, this type of growth pattern became known as Bakane, foolish seedling disease, which is a disease of rice. In 1926, Kurosawa, a Japanese scientist, discovered that the disease was caused by a fungal pathogen, Gibberella fujikuroi. By the 1930s, Japanese scientists had determined that fungus produced hyperelongation of rice stems by secreting a chemical which was given the name gibberellin. Gibberellins are normal constituents of green plants and also produced by several other microorganisms. The best known gibberellin is the gibberellic acid. In the past years of research, scientists have identified more than 80 different gibberellins and many of them occur naturally in plants. Spraying of diseased plants with gibberellin overcomes some of the symptoms caused by several viruses or molecules pathogens, indicating that the gibberellins was involved in the development of the disease. The next hormone is the ethylene. Ethylene is considered to be responsible for initiating signal in plant for wounding and senescence responses. Studies show that ethylene with another signal component, jasmonic acid, plays an essential role in plant defense responses of several pathosystems. Ethylene induces expression of certain types of PR genes and some signal components involved in defense signaling. Ethylene insensitive mutants block several PR gene expression. Ethylene insensitive mutants can be beneficial against some pathogens but deleterious to resistance against other pathogens in a specific gene-for-gene manner. Ethylene production in infected tissues can be induced. This induction is largely dependent on activation of the ethylene biosynthetic pathway in the plant tissues. Genes encoding several key enzymes involved in the ethylene biosynthesis are highly activated at the transcriptional level. It has not been shown that ethylene is produced directly by plant pathogenic fungi and bacteria. Other chemical weapons include polysaccharides, plant defense suppressors, transporters, etc. Plant viruses and viroids are not known to produce any substances themselves, but they adapt, induce and manipulate the host metabolism to replicate themselves. Ethylene is a gas that forms through the breakdown of methionine, which is present in all cells. Ethylene has a very limited solubility in water and does not accumulate within the cells but diffuses out of the cell and escapes out of the plant. Its effectiveness as a plant hormone is dependent on its rate of production versus its rate of escaping into the atmosphere. Ethylene is produced at a faster rate in rapidly growing and dividing cells, especially during the dark. New growth and newly germinated seedlings produce more ethylene than can escape the plant, which leads to elevated amounts of ethylene inhibiting leaf expansion. As the new shoot is exposed to light, reactions by phytochrome in the plant cells produce a signal for ethylene production to decrease, allowing leaf expansion. Ethylene affects cell growth and cell shape. When a growing shoot hits an obstacle while underground, ethylene production greatly increases, preventing cell elongation and causing the stem to swell. The resulting thicker stem exerts more pressure against the object, impeding its path to the surface. If the shoot does not reach the surface and the ethylene stimulus becomes prolonged, it affects the stem's natural geotrophic response, which is to grow upright, allowing it to grow around an object. Studies have also indicated that ethylene affects stem diameter and height. When stems of trees are subjected to wind, causing lateral stress, greater ethylene production occurs, resulting in thicker, more sturdy tree trunks and branches. Ethylene also affects fruit ripening. The nuclear protein ethylene insensitive 2, which is also known as EIN2, is regulated by ethylene production and in turn regulates other hormones including abscisic acid and stress hormones. The next hormone is cytokinins. Cytokinins or the CKs are a group of chemicals that influence cell division and shoot formation. They were called kinins in the past when the first cytokinin were isolated from yeast cells. They also help delay senescence of tissues. They are responsible for mediating auxin transport throughout the plant, and they also affect internodal length and leaf growth. They have a highly synergistic effect with auxins, and the ratios of these two groups of plant hormones affect most major growth periods during a plant's lifetime. Cytokinins counter the apical dominance induced by auxins. They, in conjunction with ethylene, promote abscission of leaves, flower parts and fruits. Some of the other known hormones are brassinosteroids. Brassinosteroids have been recognized as a sixth class of plant hormones which stimulate cell elongation and division, gravitropism, resistance to stress and xylem differentiation. They inhibit root growth and leaf abscission. The next one is salicylic acid. This is responsible for activating genes in plants that produce chemicals which aid in defense against pathogenic invaders. The next group is jasmonates. Jasmonates are produced from fatty acids and promote the production of defense proteins that are used to fend off invading organisms. They are believed to have a role in seed germination and affect the storage of protein in seeds and seem to affect root growth. The next group is plant peptide hormones. These encompass all small secreted peptides that are involved in cell-to-cell signaling. These small peptide hormones play crucial roles in plant growth and development, including defense mechanisms, the control of cell division and expansion, and pollen self-incompatibility. The next group is polyamines. These are strongly basic molecules with low molecular weight that have been found in all organisms studied so far. They are essential for plant growth and development and affect the process of mitosis and meiosis. The next group is nitric oxide. Nitric oxide serves as signal in hormonal and defense response. Example: stomatal closure, root development, germination, nitrogen fixation, cell death, stress response. Strigolactones implicated in the inhibition of shoot branching. The next group is characins. These are a group of plant growth regulators found in the smoke of burning plant material that have the ability to stimulate the germination of seeds. Plant growth regulators can also be synthetic substances that are similar to natural plant hormones. They are used to regulate the growth of plants and are important measures to ensure agricultural production. Plant growth regulators will not be hazardous to human health if they are used in accordance with good agricultural practice. However, if they are misused, crops will grow excessively fast, resulting in ripening on the fruit surface with the core remaining raw, which will adversely affect the palatability and the quality of fruits. Application in agriculture. The use of growth regulators produces changes in metabolism similar to those caused by certain external conditions such as temperature and length of the day. Such changes include accelerated formation of reproductive organs and intensification or inhibition of growth. Growth and organogenesis in cultivated plants are stimulated by the application of auxins and gibberellins and are inhibited by synthetic inhibitors. Such inhibitors include defoliants, which cause leaf fall, and desiccants, which cause withering of organs or the entire plant. Synthetic stimulants of auxin type, that is beta-indoloacetic acid, beta-indolobutyric acid and alpha-naphthyl lactic acid, are used to intensify root formation in scions from woody and herbaceous plants, to improve the union of tissues when transplanted and grafted, and also to prevent the ovaries from falling out in fruit and berry plants. The substances are used in different concentrations from 20 to 1000 milligrams per liter, which depend on the method by which they are applied to the plant. Gibberellins are used to accelerate the growth of seedless grapes, to bring potato tubers out of dormancy and to accelerate the fruiting of tomatoes. Synthetic growth inhibitors are used to retard the sprouting of potato tubers during storage, to inhibit the growth of cereal stems so that their resistance to lodging is heightened, and to destroy weeds. The mechanism of the inhibitory action of synthetic inhibitors has not been adequately studied. Most of the substances were found to inhibit growth by dissociating the process of phosphorylation and respiration and by suppressing the synthesis of nucleic acid. Spraying is the most common method of treating plants with growth regulators. For example, alpha-naphthyl acid and its derivatives are sprayed on fruit and berry plants to prevent the ovaries from falling out. Thus, to conclude with, plant hormones, both natural and synthetic, play a major role in plant growth and defense mechanism. Thank you.